We are going to start with the carbonic anhydrase inhibitors. Then a typical drug or the prototypical drug that I'll be using is what we call acetazolamide. Acetazolamide. So acetazolamide will be our prototype drug that I'll be using. And this drug works within the proximal tubule. Now let's come and talk about the mechanism of action of this agent. So I am going to extend the luminal side of the proximal tubule. So when you take the proximal tubule, so we have filtrate coming in here. So let's say the filtrate coming in here has sodium inside, and it also has bicarbonate ions inside. So we have sodium, we have bicarbonate, we have other electrolytes that all comes along here. And bear in mind, the proximal tubule reabsorbs 65% of the filtrate that are delivered to it, and the other part are also absorbed along the length of the nephrons. But at the proximal tubules, we have bicarbonate, we have sodium, and bicarbonate and sodium will have to be reabsorbed from here back into the interstitial spaces. Then from there they move straight into the blood again, so that they come back again, and when they come back again, the cycle will continue. The body will reabsorb bicarbonate and sodium at this point in time. Most of the time our body is constantly producing acid, carbon dioxide and other acid, and because of that the body also needs bicarbonate to help in either neutralizing or buffering or trying to maintain some acid-base balance in us. So at each point in time, our body wants to reabsorb bicarbonate to somehow balance the acid production in us, because our body is constantly producing different type of acid, like carbon dioxide and other type of acid. Okay, so now let's see what goes on over here. At the luminal side, we call this place the luminal side, and we call this place the basolateral membranes. And at the luminal side, we have a certain protein at the luminal side, and at the basolateral membranes too, we have a different type of protein at the basolateral membranes. And those proteins have different functions that they perform. And because sodium carries a charge, and because bicarbonate also somehow carries a charge, it will be very difficult for them, especially sodium, to go through the cell membrane by themselves. If you have learnt the cell membrane very well, some substances cannot diffuse or cross the cell membrane by themselves unless with the help of a transport system. And because sodium, bicarbonate all have charges, it's very difficult that they will go through the cell membrane by themselves. And because of that, at the luminal side, this is what happens. We have a pump over here. We have a pump. The name of the pump is the sodium hydrogen exchange pump. Sodium hydrogen exchange pump. Sodium hydrogen exchange pump. So what happens is that the sodium which is in the filtrate, when it gets to the proximal side, the proximal tubule, what happens is that the sodium hydrogen exchange pump will take the sodium from the filtrate and pump it into the cell. So sodium over here will come through this pump and enter into the cell. And hydrogen in the cell will also be pumped out into the luminal side of the nephron, or will be pumped into the proximal side of the nephron. So at this point in time, sodium is taken into the cell in exchange for what? Hydrogen. When the sodium gets into the cell, we have another pump which is planted on, or we have a protein which is planted on the basolateral membranes. And what this protein does is that this protein constantly throws out sodium and brings in what? Potassium. So let me just draw this thing a little bigger and let's continue. So I am just extending the diagram. So over here, I think we have the sodium hydrogen pump, where sodium is constantly thrown into the cell in exchange for what? Hydrogen. So the hydrogen comes out. And when the sodium goes into the cell, we have a pump over there that constantly throws sodium and bring in what? Potassium. So because sodium always goes out and potassium comes in, the cell, inside of the cell, is always pour in what? Sodium. So the sodium that is carried out over here is pumped into the cell. When it gets into the cell, we have the sodium potassium pump. This sodium potassium pump will constantly pump sodium out of the cell. So it gets into the interstitial spaces. Then from there, it goes back into the blood. Then it flows in circulation, while potassium is always pumped into the cell. Okay, so now once that is established, now let's move on. So when sodium is exchanged for hydrogen, the bicarbonate, which was also part of the filtrate coming, this sodium, this hydrogen that was pumped into the proximal tubule will combine with this bicarbonate. So this hydrogen will combine with this bicarbonate to form what we call carbonic acid, H2CO3. So initially we had only bicarbonate. Sodium was exchanged for what? Hydrogen. When the hydrogen comes, the hydrogen combines with this bicarbonate to form what we call what? Carbonic acid. And when we have carbonic acid, we have an enzyme at the luminal side. The name of the enzyme is what we call carbonic anhydrase enzyme. So we have an enzyme which is planted at the luminal side, and the name of the enzyme is carbonic anhydrase enzyme. What this enzyme will do is that this enzyme will split this reaction. This enzyme will split this reaction into what we call carbon dioxide and water. This enzyme will break this reaction into carbon dioxide and water. Water will continue until it gets out over here. The carbon dioxide will diffuse and go into the cell. So when the carbon dioxide goes into the cell, it meets water in the cell. So you know that inside the cell, we have what? Water in the cell. So carbon dioxide diffuses into the cell, meets water, and this time around, with the help of the enzyme, which is also found at the cytosolic spaces or in the cytosol. The name of the enzyme is carbonic anhydrase enzyme. What this enzyme will now do is that this enzyme will combine this reaction and form what we call carbonic acid. So they will combine this reaction to form what we call carbonic acid. So they will form H2CO3. So when you have H2CO3, this reaction will now split into bicarbonate, which is HCO3 minus, and hydrogen ions. So we have bicarbonate and hydrogen ions. The hydrogen ions will be pumped back again into the proximal tubule, meet bicarbonate, and this reaction will go on. But the bicarbonate which is formed will diffuse and now enter into the interstitial spaces, then from there back into the blood. So that the bicarbonate that will go back into the blood will help in maintaining what? The blood pH. And you know that our blood pH is somewhere around 7.4. And this bicarbonate, which is the base, help in maintaining what? Our blood pH. So as a way of recap, if I'm supposed to go over again, what I said is that at the proximal tubule, we have a pump we call the sodium hydrogen exchange pump. The sodium hydrogen exchange pump, what it does is that constantly it takes sodium from the filtrate and pump it into the cell. So sodium will come into the cell in exchange for what? Hydrogen ions. When hydrogen ions come, the hydrogen ions will combine with what? The bicarbonate to form what we call what? Carbonic what? Acid. And when we have the carbonic acid, then an enzyme called what? The carbonic anhydrase enzyme will split the reaction into what? Water and what? Carbon dioxide. Carbon dioxide will diffuse, go into the cell to form carbonic acid. Then with the help of the carbonic anhydrase enzyme, we are going to split the carbonic acid this time around into hydrogen ion and what? Bicarbonate. Then the bicarbonate will now diffuse and go into what? The cell or go into the blood.